Greetings everyone, I am Carl and today I am going to talk about our research study entitled
In silico docking and dynamic simulation of selected Philippine plant-derived flavonoids as SARS-CoV-2 Papain-like protease inhibitor. SARS-CoV-2 is notoriously known as the causative agent of COVID-19 disease, which disrupted the normal life of humanity. And as of June 2022, around 540 million confirmed cases and 6.3 million deaths were recorded due to COVID-19. SARS-CoV-2 is primarily composed of 4 structural and 16 non-structural proteins, or NSPs. One of the interesting non-structural protein is NSP3. NSP3 is consisted of several domains including the Papain-like protease 2. Papain-like protease, or PLpro, is a protease with 315 amino acid residues and one of the proteases responsible for producing matured NSPs. Hence, inhibiting this protease could provide a way to disrupt the activities of SARS-CoV-2. Flavonoids are one of the most abundant secondary metabolites. It is also known for its 15-carbon polyphenolic skeleton and promising antiviral activities against adenovirus, HIV, and dengue virus. Thus, we came up in an objective to provide insights into the potencies of selected flavonoids from Philippine medicinal plants as enzymatic inhibitors against SARS-CoV-2 PLpro. To achieve our objective, we perform molecular docking and molecular dynamic simulation of SARS-CoV-2 PLpro against the selected flavonoids. We also documented the pharmacokinetic properties of these flavonoids. ADME properties prediction suggested that all of the flavonoids were skin permeable and could be absorbed intestinally. All of the flavonoids have also significantly low blood-brain barrier permeability and non-toxic. Specific site molecular docking results suggested that ligands like 2, like 6, and like 9 had promising binding free energy of less than negative 7.2 kilocalories per mole. Visual protein-ligand interaction analyses show that there are 3 hydrogen bonds formed in 6WUU like 0 and 5 in 6WUU like 2 complex. Also, it enabled us to determine the amino acid residues contributed in the hydrophobic interactions which stabilize the complexes. Meanwhile, the hydrogen bonds in 6WUU League 6 complex is highly concentrated on the cormain system of the flavonoid. Conversely, around 8 hydrogen bonds were formed in the 6WUU Lig 9 complex. Molecular dynamics further validates the results we've obtained from molecular docking. The protein backbone RMSD suggested that the 3 complexes stabilizes throughout the 10 nanosecond simulation. However, the protein backbone RMSD of 6WUU League 6 indicated higher conformational changes. The RMSF suggested that the naphthalene-based binding site amino acid residues were relatively stable throughout the simulation. Meanwhile, the radius of gyration implied that the 6WU's League 9 complex was the most compact, which was further supported by the solvent-accessible surface area results. The ligand RMSD and average center of mass suggested that the ligand Lig6 moved further away from the protein's binding site. This behavior was also reflected to the ligand's number of hydrogen bonds as it dropped from 3 to 1. Meanwhile, ligands Lig2 and Lig9 maintained its number of hydrogen bonds throughout the simulation. To visualize the behavior observed from these trajectories, a simulation video will be played. This is the simulation video of 6WUU League 2 complex. As you can see here, the complex stabilizes itself throughout the simulation. Meanwhile, the simulation of 6WUU League 6 complex suggested that a flavonoid shifted its position and moves further away from the protein. Lastly, the 6WUU League 9 complex, which was the most stable and most compact, was able to stabilize itself. Moreover, MMPBSA free energy calculations provided us the free binding energies of the complexes. This suggested that 6WUU Lig 9 was the most stable, followed by 6WUU Lig 2 complex. In conclusion, we are able to document the pharmacokinetic properties of the selected Philippine plant-derived flavonoids. Molecular docking analysis suggested that cercelineol, diosmitin, and myricetrine had promising binding energies. The molecular dynamics further validated this claim and indicated that a complex formed between 6WUU and myricetrine was the most stable and most compact, while 6WUU diosmitin complex was the least stable. In vivo and in vitro analysis was suggested to further prove the potential inhibitory activities of cercelineol and myricetrine. Also, molecular docking against the human deoxycholineases is highly encouraged to determine the cross-reactivity of these compounds. And here are the references we've utilized in this study. Lastly, we would like to thank the DDOST SEI for their financial support for this study. Thank you very much and have a great day.